Liquid handling robots and related equipment have been around for several decades now. Like all fields, the tools for doing biology are under continuous development, but the robotics space for automating wet lab experiments isn't new. It's used extensively in the pharmaceutical industry, but also in high-throughput screening-based companies and core facilities at universities. Let's take a look at some of the equipment and what they do. The most basic type of automation equipment is a liquid handler. A conventional liquid handler does exactly what you would do by hand using a single-channel or multi-channel pipetteman. They employ special tips that are incompatible with LTS or universal pipettemen, but other than how they attach to the machine, they serve the same purpose as they do with manual liquid handling. Essentially, the robot is a four-dimensional CNC machine. You can move horizontally through the X and Y dimensions of the deck, up or down, and you can press the piston within the pipette head to aspirate or dispense liquid. You can control the movements of the robot across the four dimensions however you wish, but the instruments all come with software that wraps sequences of movements into commonly used operations, such as aspiration, dispensing, and mixing. Different models of liquid handlers differ in terms of the size of the workspace, which is how many plates can be positioned on the deck at one time. Whether they also include the means of moving plates within the deck, usually that's referred to as a gripper tool, how many pipette heads are accessible, and the size and volume of the pipette head. You can get single-channel pipette heads that match the size range of manual ones, from single microliter to milliliter volumes. You can also get multi-channel heads that deliver in units of 8, 96, or 384 well tips at a time. Because of the large number of tips involved, the slightly higher cost of these tips, and the need for some person to monitor and set it up, using a liquid handling instrument often gets quite pricey. This is not something to consider unless you intend to process at least a few thousand samples for most applications. There is also a significant capital investment in robotics. There is a saying that in the cost of this equipment, it runs by weight, with a price tag of around $1,000 per kilogram of equipment. So these instruments typically run in the tens to hundreds of thousands of dollars. The cost of consumables and the desire for faster liquid transfers has led to the development of other methods that do not employ tips. The most recent of these technologies are acoustic probe liquid handlers that use sound waves to move tiny droplets of liquid. These typically transfer liquid in the nanoliter scale, and larger volumes can be moved by sending more smaller volume droplets. Operationally, the acoustic liquid handler is a two-dimensional CNC machine with an acoustic probe head. That head is positioned below the well of a sample plate, and the emission of sound waves causes the droplets to jet from the plate. If another plate is positioned to receive these droplets, you can jet the liquid into a recipient cell. So basically, this instrument holds two plates face to face, it aligns the plates such that the wells being operated on are aligned, and then it jets liquid from one well to the other. The analogous operation with a conventional liquid handler would be to use a single-channel pipette head to aspirate liquid from one well of the plate and dispense it in another well of another plate. This operation is generally referred to as cherry picking. The advantages of acoustics are the minimization of reagent volumes, the speed of the transfer process, it's very fast, and the ability to dispense liquid to non-plate surfaces such as the surface of a silicon chip. The disadvantages are the cost, the newness of the technology, and some operational sensitivity to samples of certain composition. Some solvents don't work with this. An acoustic probe also lacks the ability to mix samples. It just transfers liquid. Other acoustic-based instruments exist to handle mixing. Here is an example of a device that uses sound waves beneath a plate to cause convection in the wells of a sample to achieve mixing. The image shows the movement of magnetic particles that on the left have settled to the bottom of the plate and on the right are well suspended. Thus far, the instruments we've discussed all move liquid from plates to other plates. Though some do this the entire plate at one time, these technologies all enable the cherry picking operation in which one sample at a time is moved to an arbitrary other position of another plate. So it allows n-by-n transfers of liquid. Another type of instrument, called a dispenser, does n by m transfers, where m refers to a static set of reservoirs. This instrument shown here has an m of 1. It holds a single large reservoir of liquid in one syringe, and it pushes the plunger to emit the liquid. This is a three-dimensional CNC machine. You can move the syringe left and right to direct the flow of liquid to a particular well of the plate, and you can control the depth of the plunger. Another common mechanism is to have a large bottle of liquid under positive pressure with a tube coming out of it that is attached to the dispensing head. By controlling the opening and closing of the valve in the dispensing head, the flow of liquid is turned on or off. These instruments often look much like this one in the sense that they have a 2D movement of the plate below the dispensing head. An even simpler instrument has just one dimension of movement and an 8-channel dispensing head to deliver liquid to one row of a plate at a time. Even simpler ones, such as a plate washer, which we'll come back to in a bit, dispense liquid to the entire plate simultaneously. More sophisticated dispensers are capable of delivering nanoliter volumes of liquid through the use of small piezoelectric valves that control the flow of liquid through channels. In this variant from Beckman called the Bioraptor, there are 4 reservoir channels in the pipetting head and 2-dimensional movement of the plate. You can put any amount of any of the 4 liquids into each well of a 1536 well plate in around 30 seconds. So the advantage of this method of this instrument is miniaturization of the reagents as well as a reduction in the time required to fill a plate. The plate washer is an older piece of equipment like the conventional liquid handler. They typically cost a few $1,000s and are fairly common even in academic labs. Principally, they are used for ELISA assays. You place your 96 or 384 well plate under its 96-pin dispensing and aspiration head. It can slurp the liquid out of all the wells in the plate and send it to a waste container, and it can spray liquid from a reservoir into the plate. In ELISA, having reproducible washes between the steps of the protocol is often essential for consistent data. Doing it with a manual multi-channel pipetteman with a steady hand often is sufficient, but a plate washer can handle these operations more reliably. This is something I have in my lab and would recommend having if you ever intend to do ELISA, even for a single plate experiment. Other tip-free methods of liquid handling can be done by microfluidic methods. Much like the Bioraptor, this microfluidic liquid handler can dispense the liquid from multiple channels of sample into each well of any volume of 384 well or higher density plates. More exotic microfluidic solutions are also available. In this one, you employ these plastic consumables with many reservoirs on the top. You place your samples for the experiment into these reservoirs. Below them is a network of small mixing chambers that are sealed off from one another by septa. The septa can be ablated by the instrument with a laser to open the flow of liquid from one to the next. Operationally, you program the series of mixing events for your assay by telling the instrument which septa to ablate. Many of these cassettes are stacked into a rack and then loaded into the instrument. The samples are then placed in a centrifuge that forces the liquid from the reservoirs into the mixing chambers. Finally, the results of the assay are read out colorimetrically or fluorimetrically by looking at the final mixing chambers with a microscope. For directed evolution and combinatorial experiments based on screens, there is usually a step in the procedure in which you must pick many colonies of a library and inoculate small cultures in 384 well plates. For up to a few thousand colonies, this is most easily done by hand, but if you have more than 10,000 colonies to pick, you might want a colony picker. Traditional colony pickers employed a 1-foot square petri dish. You would fill these plates with agar at a precise volume to control for the height of the liquid, and then plate your library to get nice, well-isolated colonies. Often you must plate with the plating procedure to get the density right, but once you do, the colony picking goes very quickly. The instrument takes a picture of the plate and then uses image analysis to identify the positions of the colonies. It then very quickly passes a picking head over the plate and extends a needle at each colony to pick up some cells. Once it has done this for each pin of the head, it moves the head to a plate containing fresh media and stirs the pins with that liquid. It then sterilizes the pins with ethanol and heat and then repeats the picking process. Though a colony picker can take a long time to set up, once you have it working, it picks colonies exceptionally quickly. A 20,000 colony pick will probably take about an hour. More recent instruments of this type, as shown here, work on conventional agar plates and employ more sophisticated algorithms to pick the colonies, but in the end, it all works the same. A pin is shot into the colony to pick up some cells, and then it is transferred to a plate. The large number of samples that are implicitly involved in lab automation requires the use of barcoding techniques. When working with 96, 384, or 1536 well plates, it is common to use one-dimensional barcodes in the form of stickers on the sides of the plate. When you order oligos from IDT in a plate, you'll notice that these barcodes are already present, allowing tracking. You can also buy plates already fitted with a unique barcode in this fashion. Another type of barcoding, shown here, involves special tubes with 2D barcodes on the bottom of each tube. These tubes snap into 96 or 384 well plates for storage. This labeling method allows you to segregate and track individual samples of a plate. In highly automated companies like Ginkgo or Amyris, they track their samples this way. Even in the small academic wet lab, this barcoding technology is useful for maintaining -80 stocks of cell strains. The cost of these tubes is comparable to other screw cap tubes, and the reader for the barcodes costs around $2,000. We use this in my lab for this purpose, and it is helpful in dealing with the problem of determining what is in a sample long after the experiment is completed without the need for hand labeling and the ability to read somebody's handwriting. When working with large volumes of samples and many plates, even trivial operations like removing the caps from tubes can become rate limiting. Here I show an instrument that removes the little rubber caps from the samples of a 96 well plate full of 2D barcoded tubes. Other instruments can put the caps back on. For more conventional plates that do not require single well access, you can seal and deseal the plates using tape. Here a roller of tape is positioned next to the plate and is rolled over the plate to seal it.